Max Herzog. I'm Deputy Director of Programs and Partnerships with Cleveland Water Alliance. And it's my great pleasure to welcome you today to our third and final edition of this mini series on the state of harmful algal bloom monitoring. Today's conversation is convened by Cleveland Water Alliance, a nonprofit economic development agency focused on water technology innovation and economic development, and the Great Lakes HABs Collaborative, coalition of researchers and agencies focused on communicating the science around HABs, convened by the Great Lakes Commission. We'll talk more about those orgs in just a second. Today's session is focused on innovations in nutrient monitoring technologies. So we're really fortunate to have some folks from government, from research, and a number of folks from the private sector to talk about the state of nutrient monitoring technologies and programs and where things are headed. A little bit of background about the organizations convening this and why we're putting this session together. The HABs Collaborative was started in 2015 to bridge the communication gap between scientists and decision makers regarding HABs research. Through conference sessions, newsletters, publications, and webinars, the collaborative works to improve communication through a common agenda, which is addressing science and management needs for the region. So really excited and grateful to the HABs Collaborative and the Great Lakes Commission for supporting this series. My organization, Cleveland Water Alliance, was founded in 2014 to accelerate the Great Lakes region's water economy by generating innovative solutions to global freshwater issues. We enable the development of technologies that address HABs and other water resource challenges through our test beds, innovation challenges, mentorship, and other sort of tech development and commercialization programming. So we're really excited to be able to bring sort of the technology side of things to the table for this series. I do want to note just a quick bit of housekeeping before we dive into the session. We do encourage you to share your questions via the Q&A function at the bottom of your screen, sort of the toolbar. It is distinct from the chat, so please do share things in the Q&A. It lets our panelists respond and mark which questions they've answered if they have time during the discussion, and that is where we will go to first at the end of the discussion to pull questions from the audience for our panelists. With that, it's my great pleasure to introduce our moderator for today. Katie Stamler is a water quality scientist and source water protection project manager with the Essex Region Conservation Authority. Katie, thanks for being here, and I'll hand it over to you now.
Thanks, Max. I'm really excited to be here with this great group of panelists, and I just wanted to provide a small amount of background before we dive in. And I think that probably many people on the call here are familiar with harmful algal blooms in Lake Erie and why nutrient monitoring has become such a big topic in recent history. But nutrient monitoring and analysis has a very long history in our seeking to acquire knowledge, going back even to Dave Schindler and others, looking at nutrients like nitrogen and phosphorus, trying to determine what the limiting agents are for algal growth, and just how far we've come with those technologies from doing lab analysis. And we know that even within a laboratory setting, those analysis methods have changed and become more refined. We've been able to lower detection limits. We've introduced things like automated samplers that allow us to sample during the course of a rain event, as we've learned that the bulk of nutrients move during rain events. So we want to try to capture those nutrient concentrations when we're near the top end of a hydrograph, which is very difficult for individuals to do when water is flowing very quickly. And we know, at least here in the Essex region, that tends to happen at midnight on Saturday nights, usually over the Christmas holidays. So the advent of that automated sampling has really been a game changer for people that do this type of work. And then in more recent history, we have the Great Lakes Water Quality Agreement that has new nutrient loading targets, particularly for priority tributaries into Lake Erie. But we also know that these types of harmful algal blooms are happening globally, and some of that due to eutrophication and some due to other types of disturbances. So we need to have these different and new advancing technologies that will help us get into those remote locations, help us get more reliable data on the timescale that we need it. So I'm really excited to have with us here today individuals that are working on that forefront of developing our new types of technologies that'll lead us into the future. So I'd like to introduce our panelists. So we have Colm Lynch with us, who is the CEO of Aquamatronics. Vince Kelly, who is the founder and director of Green Eyes. EJ Neefsey, who is the US Director and Chief Water Scientist of LG Sonic, although I hear he's also a Canadian, so I'm not the only one here. And Nate Manning, who is the Interim Director of National Center for Water Quality Research at Heidelberg University. So very excited to have these folks with us and really excited about the conversation that we're going to have. So to begin, I'd like to discuss the current challenges that exist in watershed monitoring agencies in obtaining the quality of data that we need and the role of your agency and the technology that you've developed that's going to help us solve those challenges. And we're going to start with Nate from Heidelberg University.
Thanks. Yeah, so the sort of monitoring that we do at Heidelberg, right, is high frequency in the sense that it's a daily, up to 3 times a day, sample. And for the big rivers, things like the Maumee or the Sandusky, that's generally fine to capture a lot of sort of the nutrient dynamics. But for the smaller, flashier systems, I think that having the automated high frequency sort of sampling that the other panelists here, their companies are looking to provide, I think is really, really important to understand sort of the dynamics of how these nutrients move through these systems, right, from the field into the creeks, into the smaller streams, into the rivers, and ultimately into the lake.
Awesome. Thank you, Nate. Vince, could you speak to us about your technology?
Yeah, thanks, Katie, and thanks, Max, for inviting me and Green Eyes to be part of the webinar. I'm the founder and director of Green Eyes, and I've been working with autonomous nutrient monitoring since graduate school a couple of decades ago now. And it's great to see it really finally taking hold in the industry. We've got more players, there's more options for users. The need is as great as it has ever been for continuous autonomous nutrient monitoring, HABs and loading estimates and everything. So I don't think I need to convince anyone of that. So the new lab that Green Eyes offers has been available since 2015. It's a wet chemical sensor. So it uses the methods that are very similar to those used in a laboratory, where we're mixing chemicals, forming a color, measuring the intensity of that color with a colorimeter. The instrument calibrates in situ before every analysis, so we can adjust. So we build effects of temperature and other things into the calibration. The reagents need to be replaced every 4 to 8 weeks, depending upon sample frequency and exactly what things you're looking at. Green Eyes currently offers units for nitrate plus nitrite, soluble reactive phosphorus, ammonia, ammonium, total nitrogen, and total phosphorus. And we've been at this for a while, and I feel pretty good about where we brought the new lab in terms of its accuracy. We produce very linear calibration curves in the laboratory. The reliability is certainly better. And where we're focusing on now is trying to make the whole operation easier for folks in the field who tend to be busy with lots of things to do other than just set up the nutrient monitors. And so where we are personally is streamlining the methods with better documentation, easier methods. And under normal circumstances, the data quality is pretty good. And the reliability has gotten pretty good. I will share with the group something that has come out. We've had an opportunity this year to deploy 2 new labs very close together on small, flashy streams. And one of the challenges that we learned is that where we mount our pump, so just in case I didn't explain that, the new lab lives in a dry sampling shed or an enclosure adjacent to the stream, and Green Eyes either supplies a pumping system or we rely on an existing pumping system at a station. We found that in these small flashy streams, we have to be very careful where we mount the pump. And it turns out if we're too close to the sediment, we could get additional phosphorus. We can even see denitrification occurring in the sediments. And we discovered this really because we had 2 instruments relatively close together. And when the stream level got very low, we saw the measured concentrations diverging some. So we've worked so hard for so many years on making the new lab be a reliable, accurate instrument. And probably we haven't focused enough time on sample delivery, which has taken over as the #1 failure point in these systems, is getting a robust sample to the instrument. And so that's where we're going nowadays. But I think with Green Eyes, some of the other patentless and other instrumentation that's coming out there, it's really come a long way in terms of accuracy and reliability. My take it's a little early for my take-home message, but I have to beat this drum all the time, and that ask users always to collect hand samples for comparison. So this just gives you more confidence in the autonomous data, allows us to correct the data. And I'm beating this drum because it's so often that folks are not collecting hand samples, or that the process to get the hand samples analyzed is months, and it's just taking longer than it really needs to. And this is another area where Green Eyes is going to try to help and focus. But those are my 2 cents right now. Thank you, Katie.
Amazing. Thank you, Vince. And I appreciate that perspective of making sure that the users actually have a good sense of how to use it when we're using new pieces of equipment and trying to figure out where to put it and how to use it. That's having that at the forefront is really important. I'm going to move over to Colm, who's going to give us a different perspective, as he's not in the US or Canada. So he's going to give us a more international perspective.
Yeah, thanks, Katie. First of all, I'm very grateful to be participating in this event. And I suppose to bring a different perspective on it in terms of an international perspective on the formation of harmful algae blooms. The first thing I'd like to say is that it really truly is a global problem. It's one that we're talking about in the Great Lakes, but one that we're experiencing across the globe. So we are a company based in Ireland, and we'd like to pride ourselves on high levels of water quality, but we do have harmful algae blooms forming in some of the lakes which are pulling our drinking water from. So we're experiencing the same challenges. Our company, HAB Monitoring, has about 25 years plus experience in water quality analysis. So we're a bunch of chemists, and really our background is that the whole focus on analysis and accuracy. And we developed a suite of analyzers, and the first ones to market are nitrate and nitrite analyzer, and a slightly different take on technology. We use ion chromatography coupled with UV LED detection to be highly selective and highly accurate. And our initial kind of development of this product was to go into the environmental monitoring market, and we worked with the US EPA in the early stages of our product development. One of the challenges just in terms of getting through to that environmental monitoring sector, of course, is funding. So it's not a big market yet for our company, because the wastewater industry has got a bigger budget and a bigger pull in terms of where you deploy. But just looking at the products we have here on the screen, on the far right, we have a deployable instrument which can come with a solar panel and be left out for long periods of time for collecting data. And that picture on the right hand side is in Ireland, and on the bottom left hand side, by contrast, we've got one in Tasmania and just off Australia. And one of the cleanest water courses I think you will ever see on the face of the planet, and they do grab and lab, to make sure that everything is okay. But they put a instrument on a river and literally up and down the river testing the water quality. And in real time, they found a huge variance between what their grab and lab results were taking versus the real time analysis. So I think one of the key things and the challenges that the agencies have in terms of, getting the data is, first of all, we need to benchmark to establish exactly how big the problem is, where the problem is, because we can probably all run to kind of suggest where we think the source of nutrient influx is coming from, but probably need to baseline that in the first instance. And in some cases where you think exactly what the water quality is, where the sources are, you may be proven incorrect. So our technology is pretty accurate. When I say pretty accurate, it is highly accurate. It's effectively a laboratory level accuracy in the field. And we have developed sensors that we can deploy in remote places to monitor water quality in the areas such as we're describing. And yeah, so that's just a very brief introduction to us.
Amazing. Thank you, Colm. And we'll move over to EJ Neepsey.
Thank you, Katie, and thank you for inviting us to participate. So at LG Sonic, our heritage is data-driven ultrasonic algae control. So we use water quality parameters to really select which ultrasonic programs we use to push harmful algal blooms out of the photic zone in the water and make the water cleaner and safer for everybody. But we always want to make sure that we're able to predict as far into the future as possible. And so we're always interested in looking at nutrient dynamics, which is really, setting up what that nutrient budget is going to be that the algae can use to do business. And so we landed on orthophosphate as phosphorus for our primary target for nutrient monitoring, because that's the fraction that's really the most bioavailable to a lot of our target freshwater species. And so we decided to partner with Dublin City University in Ireland to develop a sensor lab on chip that's based on the yellow method. So it has the benefits of being a pretty standard colorimetric method. And the fact that it uses the yellow method means that the reagent is a lot more durable. It can be stored in a lot more realistic and pragmatic conditions, such as, storage sheds that, might get to pretty high temperatures. And our customers can order a lot of reagent at a time, so they don't have to worry about, expiration within an unreasonably short period, so they can get ahead on their materials. But we've deployed it on a vast variety of conditions, from snow-lined creeks in Ohio all the way to estuaries in Ireland. And to get back to Vince's point about the importance of grab samples, it is critical. And we have done some cross-checking with lab and bench sampling, and it's really bang on within the range. So we are able to collect at our standard intervals about 3 samples a day, but it is proportional to reagent use, so the user is able to collect a lot more frequent samples if possible, and that allows them to get much better information on what their nutrient budget is, and really where they should focus a lot of their efforts in nutrient management.
Awesome. Thanks, EJ. It's really, really incredible to see where you guys have taken technology, and I know you're just a subset of people that are working on this type of thing. But of course, that progress always comes with challenges and limitations. So I'd like for us now to discuss some of the different challenges that you've faced with implementing the new technologies. And Vince kind of started alluding to some of the installation parts of things. But Vince, I'm going to turn to you to talk first about any other challenges that you're facing with using the technologies.
Great, thanks, Katie. The biggest challenges that we see are just having folks understand that these are not optical, well, and there are some optical nitrate sensors out there, but when we're talking wet chemistry or ion chromatography, it's a little more complicated than sticking a multi-probe in the water. But having said that, it has advantages. So we can use onboard standards for calibration, which is something that most optical and electrode type sensors cannot do. So it's a bit more complicated, but we have better tools for evaluating accuracy and performance. And so what I identified some years ago was just trying to reduce the demands and time required in the field for folks so that these could really achieve broad deployment, broad acceptance, similar to SONs or dissolved oxygen type sensors that also have some of their own inherent challenges. And so for us personally as a company, it's about improving our documentation, improving our methods, and improving the training of customers in the field, and just trying to whack-a-mole failure points. Like this, there's simple things you probably saw in the photo, there's a lot of pinch clamps and things. So there's a number, you just have to have attention to detail and good documentation and procedures to help folks handle that. I think I mentioned challenges with sample delivery, and I would say I came from an oceanography background, and we sort of thought of freshwater and estuarine water as easier to measure because often the concentrations are higher than open ocean work. That was a lie, because it turns out there are a lot of interferences in these flashy freshwater streams just when we really need the data the most. So just at that Friday night at 11 o'clock when there's a big storm, we also got a lot of sediment with it and other interferences that can become a challenge. And so I think Green Eyes is handling that. We have a lot of tricks up our sleeve and how we handle that. I'm sure Colm and Nate as well are evolving or have evolved with that. But I think these are, typical growing pains for a still relatively young industry of field monitoring of nutrients. And that I would say that we've done quite well. But so the number one challenge is for just customers to realize it's not a multi-probe, wet chemistry. We're just going to need to spend a little more time in the field and a little more attention to detail, and maybe we keep an eye on it a little bit more. And in the end, if we do those things, we can actually have higher accuracy and understand what we're measuring even better than an optical or electrode-based sonde. Thank you.
Awesome, thank you. And Colm, when we met earlier, you had also raised a different challenge with regulations and stuff. Would you be able to speak with that?
Sure, yeah. I suppose the first thing I'd say, I'd echo what Vince is saying in terms of the technical challenges. I think we're all facing the same thing in terms of getting over some hurdles, and we're going about it in different ways. And because obviously we have different technology platforms, and so I definitely would identify with that. And I would also identify with the commercial challenge as well in terms of getting analyzers out there. That's probably the biggest challenge. One of the things that drives, let's say, traction in the marketplace for environmental monitoring, especially in Europe, is underpinned by regulation. So where you get an authority where there's regulation and consent on discharge licenses, which is also policed by the EPA or whoever the local authorities are, you then get people who will look for compliance, and compliance will drive monitoring. And this is how you get your monitor out or your analyzer into the field. There is a number of ways that then, in terms of who are the end user for the analyzer. So whoever is discharging into our waterways and maybe potential sources of this nutrient ingress. So which could be activities coming from wastewater treatment, also coming from maybe agricultural runoff, and things like this. And in some cases, it can be regulation. In some cases, it can be training. And in some cases, it's just working with people to get them to understand the impact of maybe something that they're doing, that they really don't understand what the net effect of that is on the collective of a water body. So I think one of the challenges we need to do, as a community, and I'm talking as a global community, is to really look at that regulation and temper the regulation and balance it in such a way that people don't see it as policing it or, kind of nanny state sort of thing, as opposed to bringing people on a journey with you so they understand the impact. So I think if I can speak to Europe for a second, probably the leaders in Europe in this area is the Netherlands. I'd love to say it was Ireland, but it's not. It really is the Netherlands, and because of how close they are to seawater level at land in the Netherlands, I think it's something like 90% of their drinking water is sourced within 3 meters of the surface. So they are very cognizant of nutrient levels within their waterways. So as a society, they push for the need to measure, control, and therefore reduce at source your pollution root cause. So I think that is one big constraint in terms of getting these technologies out into the field. But if that was removed, you still need to overcome the challenges that Vince spoke to. So, you know, in the different water matrix, how can the analyzers overcome what's out there? How can you deploy for long periods of time without interference or without having to go to service in these remote areas? So there's a number of challenges there, and I think individually we are all quite a long way along the road in terms of resolving those challenges. But I would say, as you mentioned, Katie, probably regulation, if it was pushed from the top through regulation, I think it would remove an awful lot of inertia in terms of getting this data.
Yeah, thanks, Colm. And really, you know, thinking about myself and my team and the grab samples that we take and who those individuals are, if you put that equipment out, it isn't just a sonde, and which is complicated in itself. It isn't just filling a bottle. So you do need to have that more technical expertise on the ground locally to operate those things. And I think that's important to know as well, that, you know, it doesn't take the place of the individual who is going out and getting to know the stream and the system. It's that person is still there doing that work. So that's really like that as well. EJ, I'm going to turn to you to talk about national water monitoring plans.
Yeah, absolutely. So one of the things that is definitely going to be needed going forward in the United States and Canada is, you know, a national nutrient monitoring program, so that we can, you know, understand, you know, what our nutrient budget is at the various, you know, stream reach levels. And this will allow us to really optimize the practices that we put out in the field. So it could be a combination of, you know, nutrient reduction at the paddock scale for our producer community. It can be looking at some green infrastructure in urban communities to take some of that impervious surface runoff and be able to capture it before it ends up in our rivers and streams and lakes. But we're going to need to have some data to be able to monitor and to gauge the effectiveness of those practices, so we can right size them and, you know, make on-field edits as we go forward. And another important part of, you know, really linking sensor data with action is make sure we're working with our regulatory communities so that they understand, you know, exactly how our technologies work, and so that we can, you know, incorporate that into their data governance and data quality plans, so that we can, you know, build confidence in what our technology is reporting, and so it can, you know, really help them make the decisions they need to make our water safer and cleaner and better for everyone to use.
Yeah, that's a good point too there, EJ. There, I know there's always all the conversations about even inter-lab comparisons. And did you have your sample analyzed at this lab or this lab, and is it comparable? Did you filter on site? Did you not filter on site? So yeah, having that sort of standardized information so that people trust the data and have an understanding that it is just as reliable as lab data. So I always like to end things on a high note and talk about, you know, hopes and dreams about the future. So I'd like to talk about you know, what the future of nutrient monitoring might look like. And we're going to start where we ended last time. We're going to start with EJ.
Yeah, so really our idea of the future is we want to make sure that our sensors are smaller and tougher, so we can make sure we're having more data in more places. And it also means integrating those with digital twins so that we can create products that are useful to the customer, which is really the public.
Amazing. Thanks, EJ. And Nate, you're the guy that's on the ground doing lots of the monitoring. So we're gonna end with you on this one.
Yeah, you know, I think the National Center for Water Quality Research is in sort of a unique position to talk about sort of what might be needed in the future, right? So we have a data set that goes back 50 years. We have lots and lots of data, and we've done lots and lots of different collection techniques. laboratory techniques. We've deployed some of the sensors that, the other panelists have developed, right? So we have, things like the new labs out in the field. And, I think one of the things that we've sort of come across, right, is the length of deployment for these things, right, between service intervals, right, needs to be long enough that you're getting meaningful, sort of patterns in the data, right? Being able to pick up trends. A 3-week deployment is probably not enough, right? You need to have sort of an entire season at the minimum, right? Making these things, a little more portable, maybe a little more, easier to deploy. And a lot of them have become that, right? Like over the years, we've seen a lot of the improvements that these deployable sensors have made, and they've made big improvements. And then, one of the other things, we're sort of spread out across the state, right? When the lab started 50 years ago, there were, 2 rivers that we monitored that were nearby the lab. We now monitor, 23 sites across Ohio and Michigan. And so having the ability to, through telemetry, interact with these sensors from a remote position, right? To change, it's sampling, the time between samples remotely, or, to check on, the levels of the different chemicals in some of these wet labs, would be great for, an organization like ours, where we may have these things deployed to a place that, is a 6-hour drive, round trip, and isn't logistically easy to get to. And so I think the automation of these, the chemical testing, as well as sort of the ability to interact with them at a distance, right, I think is really important moving forward.
Yeah, I agree. Anytime you can interact with your equipment at a distance is really, really handy. I always say it's a game changer when we got modems for our ISCO auto samplers and that sort of thing. And we have some new turbidity probes that we're using that have to be charged back in the office.
Right.
So, that kind of, advancing the technology so that there's a swappable battery or something.
And that adds up to gaps in your data, right?
Mm-hmm.
And one of the things that we do in lab is, really analyze our data and look at patterns in it. And what we found is, you really have to have high frequency, like, just a weekly sample isn't going to tell you much, right? If you're doing weekly sampling, you also have to have the storm sampling, right? To be able to capture those events, and really be able to differentiate between the rising and the falling limb of these storm events. And I think, these sensors are getting us there. And I'm excited to see, the advancements in them over the coming years.
Yeah, absolutely. Well, thank you all for that part of the formal panel discussion. And we'll move over to the Q&A. So I think you guys should be able to see them as well, but I'll read them out, and then you can, if there's a question that you feel like you would like to answer, please feel free to jump in. So the first question up there is, when you design a remote analysis system, what is your target service interval?
So I can jump in on this one if that helps, Katie. We've got a number of different intervals depending on where we're deploying. So if you're going to go to a very remote area where access is not easy, for example, we're doing something in Spain at the moment where we're using solar panels to power the instrument, which is much easier in Spain, as you can imagine, than other places in terms of hours of sunlight. We're looking to go there about once a month, which Nate was saying, might be, you might need longer than that. But so if we can get somewhere between 1 month and 6 months is actually the kind of the window that we're looking to do the the service interval at. The service interval is driven by the number of data points you want per day. So I would say that, and if you're taking maybe 3 to 4 data points a day, 6 months is easily achievable. If you want more than that, so it's trying to get into the mind of how many data points is enough. So if you look at the wastewater industry, they might want it every 15 minutes or every hour, but maybe from monitoring on an environmental impact scale, maybe every couple of or 2 or 3 times a day might be sufficient. And if I could just use one example in terms of that, in Ireland, we deployed, an agency deployed to try and identify a source of a contamination to a watershed that was leading into an estuary. And it was coming from agriculture. And how they were able to identify what was going on there was they took samples 4 times a day. But what they were looking for was not just the nitrate in terms of the nutrient, because if the source of the nutrient was coming from an agricultural runoff, it could be from a fertilizer, or it could be, and I see one of the questions that Sandy has put in the thing, is could it be from manure, which is so, and if it's from a manure type source, you're going to get the formation of ammonium, and then you're going to get that oxidizing to nitrite and then nitrate. So the beauty of the analyzer that we have, it'll pick up both of those. And if you're picking up nitrite, that is an indication of you have a runoff from a manure type source and it's somewhere upstream. And depending on the level of that nitrite, because it's highly labile, that it can tell you how quickly it's happened beforehand. So I would say answer the question in 2 parts. Is one that you would look to maybe do probably 3 to 4 samples a day, deploy for 6 months without servicing, and then look at the type of analysis that you're getting to identify the type of pollution event that's coming downstream.
Cool, which I think, Colm, you kind of got into I'll go to you as well, Vince. The second part of the next question that was how do you use the technology to start identifying sources? But yeah, go ahead, Vince, to jump in.
Yeah, I'll just be quick. At Green Eyes, so we would set a minimum service frequency of 4 weeks. And currently, our instruments sample between every 1 hour and every 4 hours, typically 2, so 12 samples a day. And that normally gets us 6 weeks, but we can push that, we can drop the frequency and often get to 8 weeks. So for Green Eyes, we're in that 4 to 8 week range on service intervals.
What's involved with service intervals, like having to change out reagents and that kind of thing?
Yeah, and so on the new lab, it's these bags of reagents that have to be swapped out, and with cleaning of some of the inlet tubing, replacing the filter that you see. Sometimes you have a sample chamber, we might need to clean that out. And nothing's particularly challenging. It just takes some attention to detail. We'll run some cleaning solutions through it, make sure the mechanical parts of the syringe is still sealing and things like that. So on a single channel new lab for reactive phosphorus, that servicing might only take half an hour. One of our really complex systems, it could be 2 hours.
Okay, cool. So the next couple of questions come from Sandy Ben and about more of the if we can use the technology to help us identify sources of nutrients. So whether that might be at a smaller scale or at a watershed scale. Anyone want to jump on that one? You can also type the answer if or we can say, ooh, that was a hard one.
Yeah, I mean, really, just to put in my 2 cents, it really depends on the spatial scale and where you're installing the sensors. So if you're really getting down to a stream reach, which maybe is a couple 100 meters to a kilometer, maybe you can get into that neighborhood of identifying sources. But if you're integrating over, maybe a larger river, it gets a little more difficult. You're certainly into that non-point source domain. And of course, when we're doing this, we want to make sure that we're taking a collaborative approach with all of our abutters and all of our producers. And I mean, I know at least from a fertilizer perspective that I would like to be told if I'm wasting money. I never really like to spend more than I need to. So there needs to be a collaborative approach with all of our land users in the community.
Great, thank you. Go ahead.
on that same sort of thought, so I had mentioned before being able to distinguish between the rising and the falling limb of a storm event. That difference, the hysteresis loop, right? It'll change over time, and we can measure that change, right? And so if you have super high frequency, right, every 15 minutes during a storm event, one of the things that can tell you is essentially how proximal those nutrients are to the river, right? How close are they? Are they coming from overland, surface flow? Is it subsurface flow? Is it, sort of deep sediment flow? And so having these deployable sensors, right, that can sort of kick in during a storm event and give you that, 15-minute interval, I think will help with sort of understanding sort of where in the landscape these nutrients are, how long they're taking to get into the rivers, and the pathways that they're taking, right? Are they ending up in field tile and being rushed out to the river, and those change, right? That loop, the shape of that loop, the direction it goes, how complex it is. And so I think that's something that is going to really help, that these sort of sensors can really help us get a better understanding of.
Yeah, I like that. Yeah, tying it to event sampling would be very interesting. But go ahead. Actually, Colin, did you want to say anything? I know you.
Well, it's a really good point by Nate, and I just wanted to say that. So the way that we would get around that is that we have an IoT platform, so everything's pushed to the cloud, and you're sitting at home in the comfort of your living room in the middle of a storm event, and you can actually from there then change the duty cycle of the analysis to every 15 minutes for the period of time that the storm is outside, and then you can choke it back again, whatever you need to do to adjust the frequency, and that can all be done remotely. And also the data then is presented to you on your PC or whatever at home. So yeah, that's definitely doable.
Nothing better than sitting on your couch on the weekend, watching a movie, dialing into your equipment. Never not working, that's what I say. Go ahead, Vince.
Yeah, thanks, Katie. The thing that I would add is, this is commonly the case, is more parameters are better than fewer parameters. And so now customers and monitoring folks are having more options for different instruments. The number of the different nutrient species that you can measure is growing. And then it's really nice to have traditional water quality data to go along with. So you can relate nutrient pulses with turbidity. And as Nate was talking about, sometimes you can see the initial overland rush of nutrients, and then you can see the subsequent groundwater flow of nutrients at your site. And it helps to have some basic water quality data to interface with that. So anytime you can add the traditional sensors to nutrient sensors, or the nutrient sensors to your traditional monitoring network, it's going to help you unpiece, put that puzzle together.
Well, it's sort of related question. So the real-time versus continuous, and the ability to download later as you develop the technology, how do you decide what's needed and what's offered? So I think, yeah, similar to that, like how quickly does the analysis happen? How quickly is that kind of posted? Are you downloading it on the regular? So I think that's maybe what the question's getting at.
I think for us on that one, Katie, our maximum duty cycle is every 15 minutes. So that's the maximum we can produce a data set. And the minute that the analysis set is done, it's pushed to the cloud. So the data is available instantaneously. You have many options of communicating with the sensor, but probably for remote monitoring, that's probably the neatest. But you can go to the actual device itself. You can pull data off it. You can download and CSV files also from the web portal. Mm-hmm. So I would say accessing the data that's not an issue.
Cool. Question, are the analysis standards an impediment to the sensor and technology innovation? So I think the actual reagents, is that part of what's a challenge?
Or perhaps maybe are we talking about, oh, that's a good question. Yeah, it's the analysis standards. You mean in terms of the laboratory accreditation standards, or the, yeah, maybe there's two parts to that question. So there's one part of it is if you're using this data in terms of a regulatory compliance, maybe the question is, what standard is the data going to stand up to under scrutiny? From our perspective, we don't have standards on the analyzer. It is an ISO accredited ETV analyzer, but it doesn't conform, let's say, to what you might find in an ISO accredited laboratory. That's not something you would typically find with an analyzer.
Okay. Yeah, and I think that kind of goes along too with a question there that's compared to traditional lab analysis. How sensitive are they? So if anybody else wants to answer one or both of those. Go ahead, Vince.
The sensitivity is generally comparable. And the challenge becomes that we're taking methods designed for a laboratory with controlled temperature and filtered water. And we're having to go outside the box on that and handle different things. And so in general, it's good enough if we're going to talk in milligrams per liter, where I think everybody's technology is probably better than 0.0, in that 0.01 milligram per liter area. It's certainly achievable. But I would also add that sometimes we have to do some, our real-time data, we call it preliminary, because there can be post-data processing. And if we have a temperature effect that we want to correct for, this summer, in fact, we saw it, temperatures were, 100 degrees. That's not a stable 20, that's not 70 degrees that we're working in the lab. So it can have some effects. Again, these are tractable problems. Absolutely.
Yeah, I'd just like to add that from an R&D perspective, the standards really do give us a target to hit. So it's a massively good thing when we're trying to, develop a technique that's going to be, comparable with existing standards. So that's definitely helpful. And also, just in terms of, comparing to, traditional grab sampling, it really does help us make the case that, it's within that range, and you do get a lot more data because there's a lot of events that you'll be able to capture that really do have a management implication that we wouldn't if we're just going out for our usual monitoring program using jars and people. So that's definitely helpful for us.
Yeah, I would add to that. We have our own in-house analytical chemistry lab that does all of our sample processing and stuff, but we do deploy these sensors in certain places, and I would agree that for the most part, their results are fairly similar to what we're getting, out of the lab. The one thing that I would mention is that you can often see sort of, I don't know if degradation is the right, but there's a service interval involved with these, and so if there's high sediment in your system or something, you can see sort of some fluctuation in the data that is related to those environmental conditions that change. But as a general rule, I do believe that these sensors are reasonably close to what you would get out of a laboratory setting.
Awesome. The last kind of technical question here is if we can describe how the new lab or the other technologies, TP and TN tools, digest the aerobic component of the parameters. Did I say aerobic? I meant organic.
Yeah, organic. Yeah. I was aerobic. Yes. So when we developed the total nitrogen, total phosphorus method, we tried to emulate the Jorge Valderrama method, where we're doing simultaneous oxidation of nitrogen and phosphorus to nitrate and phosphate. So this is a 30-minute digestion at 120 degrees C with a basic persulfate solution. And so again, and this is what I think all the manufacturers, we want to have comparable data. So we're trying to follow established methods on how we do things. And so it takes a while. You know, this is not the digestion is 30 minutes for green eyes. You know, we have 10 minutes of heating, 10 minutes of cooling. That's 40 minutes, another 5 minutes of flushing and mixing. You know, we're really there's no way we could do it any faster than once an hour. And to be honest, our TNTP units sample at along with nitrate and phosphate data every 2 hours, really the most reasonable, fastest method. But we are able to pretty much emulate the laboratory digestion, and we're now doing that on solar power.
Cool. Does anybody else want to address that question at all?
Yeah, at least in terms of like what we're looking at orthophosphate. So that's going to be the fraction that's available to drive algal blooms, and it's going to be dissolved into the sample and into the environment. So we use EPA standard filtration to, you know, exclude any of that solid fraction. And so, you know, because we're looking at orthophosphate, we eliminate the need to do any sort of digestion.
When you're filtering the samples, like I'm thinking about our streams that can get pretty full of sediment pretty fast. Like, does that potentially mean that your filters might need to be changed more frequently if you're in a high sediment area?
Yeah, I mean, if you definitely have to, you know, tailor your service intervals to your environment. And so if there's more turbidity in the environment, then those filters are have to be serviced more frequently. So that's just a basic cost of doing business to get the high quality data that we all need.
Mm-hmm. Well, that's the last of the questions that's in the Q&A. So I don't know if anybody else, any of our panelists want to have, you know, anything that you missed that you would like to say now, or and otherwise I'll hand it back over to Max when we're ready.
There's just one other thing, maybe just very briefly, Katie. Angel asked the question, I answered it by typing
yes.
into a thing, but it was just regarding the salinity. And it's a really good question because that's quite challenging. Just very briefly, our analyzer just operates on sodium chloride as a non-hazardous eluent, and this just enables us to move from fresh water to fully saline water without any need for recalibrations, which is pretty unique. But yeah, so if anybody out there is working in a saline challenging matrix, yeah, that's something that we can take care of.
Cool. Yeah, go ahead, Vince.
Yeah, I just want to say thanks for the folks that joined the meeting, because this is, I apologize if this is a bit of a sales pitch, but in truth, we really need to make these measurements. Like, so everyone here, all the panelists, and probably everyone that has joined this webinar understands that nutrient pollution is the #1 driver of water quality degradation. And we don't measure it at anywhere near the frequency, both time and spatial coverage, that we really need to understand the problem. It's an exciting time for us and for me. I've been doing this stuff for some time. There's a lot more opportunities to measure the efficiencies of nutrient remediation procedures, wetland restorations, and it's an exciting time for users because you can be on the leading edge of helping the society make these measurements and improve them. Green Eyes relies on our customers an awful lot to improve these methods. And so, you know, this is a movement that's happening. Thanks for joining the webinar. And we, you know, we look forward to hearing from you. And now that there are more quality options out there for autonomous nutrient monitoring, it's super important, and it's an exciting time.
Thank you, Vince. That's a great closing. So thank you to our panelists for taking the time and being here, to the audience members who showed up and listened and asked good questions, and to the HABs Collaborative and Cleveland Water Alliance for making this whole webinar series possible. So I don't know, Max, if you have anything else to close with, but otherwise we'll close here. So thank you for joining us.
Yeah, I'll just echo the thanks, and thanks, Katie, for moderating today. If folks want to review the recording of this session, it will be shared by email to you later. And if you want to go back and look at the last 2 sessions, one focused on technology for harmful algal bloom monitoring and one focused on the use of the data for harmful algal bloom management and nutrient management. Those are available on Cleveland Water Alliance's YouTube. Encourage you to sign up for Great Lakes HABs Collaborative and Cleveland Water Alliance mailing lists to stay abreast of future, informational opportunities. And yeah, thanks so much for being here.